on your Google Drive. I'm going to share my screen here. Okay, so this should be your Google Drive. If you have access to your Google Drive now, if you can see the Google Drive, let me know. Type 1 in the chat. All right, so we have 3 files that we are going to download, and I've posted those files on the chat. So the first one is here. We are going to download the RNA-seq. It is a zip file. Please download that file. And then the other 2 files that you're going to download are these ones here. Saccharomyces, the .ff file and the .gff3 file. So these 3 files are the ones we are going to download. Somebody's not seeing my screen. If the rest of you are seeing my screen, just type 1. It may be a network issue. It may be a network issue if you cannot see my screen. Okay, so there are people who are seeing my screen. Please check your network and download those 3 files. There are 3. RNA-seq, the Saccharomyces files, there are 2, 1, 2, and then the third one. Download them, and then we are going to move the files into the bioinformatics folder. We are going to move the files into the bioinformatics folder. Please pay attention. When you download the file, pay attention. Mine are in the downloads. So when you come to the downloads, you should have the 3 files downloaded, just like you can see here. You should have the 3 files downloaded, depending on where yours is going. So what I want you to do is to open the zip file, this one here, and then copy the RNA-seq and put it in your bioinformatics folder. Okay. Right click and paste it there. Now I'm not going to paste mine because it's already right here. So please bring the folder, okay, not the zipped folder, open it and bring the folder here. And when you open that folder, you should be able to see the different files that are inside here. So in your bioinformatics folder, you will bring the RNA-seq folder here. Now, remember, there are also other 2 files that you downloaded, the Saccharomyces. You're going to copy both of them. And when you come to the bioinformatics folder, put them inside the RNA-seq folder itself. Okay? So you can paste them there. Mine are already there. I'll just probably replace them, but they're already there. So inside your RNA-seq in the bioinformatics folder, you will have all the other files that are in there, including the 2 that you have downloaded separately. So whatever we are going to use today will be inside the RNA-seq folder, which will be inside your bioinformatics folder. All right, so before we open these and see which files we are going to use today, let me know if you have successfully downloaded or if we need more time to download. In the chat, type 1 if you have downloaded the 3 files from the Google Drive, and if you have put the RNA-seq into the bioinformatics folder. Okay, so those that are typing 0, please explain if you are still downloading or if you have got a challenge. Did you extract the RNA-seq folder? Yes. Just on where you've downloaded, it should be a zip file. Just open it. You don't even need to extract copy the RNSC folder from here. And then place it into your bioinformatics folder. Okay, the link to the drive. Stephen, you can share the link to the drive. Okay, so we are giving a few more minutes for the rest of us to download and place the file, to download and place that file into the bioinformatics folder, HTTP error. It may mean that your internet is slow. We should be able to download the files that are needed, the 3 of them. This one and the RNA-seq. Okay, and when you download, take them to the Google Drive, but make sure that the .ff file and the .gff3 file are also put inside the RNA-seq data. Okay, so it looks like we have successfully downloaded the files that we are going to use today, and we have managed to place them into the bioinformatics folder. We shall later see how to make the RNA-seq folder our working directory. For now, we are going to install some packages that we need for this particular session. So on your bioinformatics folder, inside your bioinformatics folder, on your desktop, this one here, in that RNA-seq, this one here, that we have just placed into the folder, open it up. You will see different R files that we are going to use. The first one that contains the packages that we are going to install is this one here, RNA-seq packages. Now, you can open the R file from here by double-clicking on it. And it should open in RStudio. And it should open in RStudio. So I would like you to open your RNA-seq packages file so that we can all see it in RStudio, So I'm going to pause here just to find out if we are on the same page. And okay, when you open it, just type 1. If you want us to repeat, we can also do that. Okay, so I think, okay, so I'm going to do this again. I'm going to go through what I was just saying again. So I'll close my RNA-seq packages file here. Now, what I said was in your bioinformatics folder, okay? On your desktop. So let me just go back to my desktop. So this is my bioinformatics folder. And by now, we have downloaded and we have the RNA-seq folder, which is inside here. The files that you're going to use today are inside the RNA-seq, So you will open the RNA-seq and we are going to install packages. The file that contains the packages is the RNA-seq _ packages. So this is the file. Double click on the file and then it should open up in R. Okay. Okay, so if you've opened this successfully, these are the packages. They're not so many, so we are going to install them. But I would like you to run these one by one, one by one. So we'll start from the first line, move to the next line, and allow it to install. I mean, I already installed mine, but run them and allow them to install. Okay. As you're installing, it will ask you to update all, some, none. If it does, you update all by typing A down here in the console and then press enter and then allow it to complete the installation. Allow it to complete the installation. So when you see the red button, that means that the installation is still happening and you have to wait until it's done and then you move to the next package. You move to the next package. So right now, we are installing the first one. Okay. So this is the process that you'll be going through. And you install, allow it to complete, and then run the next one. Okay, so if you are seeing what I'm seeing, or if you are successfully installing the packages, type 1. If you're having challenges, you can describe the challenge in the chat. So 1, I see 111. It means we are installing the packages successfully. If you're having a challenge, you could type an error or something that you're seeing. Okay, so yes, we are all still installing. If you are having no challenges, continue installing. So when it's done, it should bring this statement here. The downloaded binary packages are in that. Then you move to the next one. The cursor will have moved already. Okay, install the next one as well. Run the next line as well. Okay, somebody says, please repeat. All right, so when you open, and I want to believe that the repetition is for the installation. So when you open the RNA-seq packages R file, it contains the packages that we are going to install that we need for the RNA-seq analysis. Okay, so we are going to run these, but we're going to run them one by one. So you start with the first line, okay, and then click run. Okay, that is done. Then click run to install the Bio Manager. Okay, then when it's done, now this line here will not install if it's already installed. That's what will happen. So move to the next one, install. If it asks you to update, down here, when you see this prompt here down in the console, prompting you to update either all or some or none, just type A for all and then press enter. Then it will continue updating. It will continue updating. And it's going to do that, I think, for all the packages, these ones that you're seeing here. So keep running them until they are done. Keep running them until they are done. So if that one has been completed, then we'll move to the next one. The cursor is already in the next one, which is our subread. Run as well. It should be doing the same thing. If it asks you to update, type A for all, press enter, and proceed. Allow it to download until it's done, and then move to the next package. Okay, so that one is done. Then run the next one until you're done. Okay, we'll give you a bit of time as we check in the chat. Can I install at once by highlighting? All right, so in this particular case, we tried it. What happens is you might fail to update all the packages. So you might end up making an update for only one of them, and then they will not be installed. So please install them one by one so that we know that you have successfully installed the packages. Okay. If you've installed, please install the bioconductor packages and also the CRAN packages themselves. So the PV class as well. Also run that, do that installation. I already have them, but you can just install. It should take a very short time. And also this other CRAN package, which is the P heat map as well. Yeah, so it's good to start running your code from line 1 just to make sure that the installation is successful. You might run into an error later on when you're trying to load the libraries, and then some of the packages were not installed. Some of the packages were not installed, especially when you run a block installation for these. So just do them one by one so that we are sure that the installation has been successful for each of the packages. For each of the packages. Okay, so warnings are okay. So if it gives you that, then you need to um, update each and every one of them. All. Okay. If you're getting warnings, that is okay. If you've successfully installed all the packages, type 1. If you've successfully installed all the packages, type 1, still running. Okay. Confirm if you have installed the packages. Ensure that you have a good internet connection as you install the packages as well. Okay, it's still running. How can you update? When you're updating the package down here, if it prompts you in the console down, here in the console down, type A and enter. Just type A and enter. I'm probably going to enter, but it's going to give me an error because I typed A without having a prompt to do that. But if it prompts you, type A and enter. Type A in that console down there and enter. So that warning, package is not installed when the versions are the same or greater, is okay, because if you already have the package, it will not reinstall it. If you have a package, if you have that package and it's of a higher version than what you have, it may not reinstall it. So it means you already have it. Now that is okay. That is fine. So continue running and installing the packages. Continue running and installing the packages. The functions that we are going to use going forward will require these packages to have been successfully installed. So it's important that you continue running and installing each of them successfully. Warnings are okay, but errors are not okay as you do the installation. Errors are not okay. It probably means there's a challenge in the installation process. Okay. We give a few more minutes, about 3 minutes, and then we can start the presentation. And then we can start the presentation. Okay, somebody says they've installed. I want to believe that this means you have successfully done the installation for all the packages that we have in the RNA-seq packages file. Okay, if it's still running, allow it to continue running. You need more time. Yes, you have the time to continue installing. You do have the time to continue installing. Okay, we intend to start in about 6 minutes. How about the bioconductor packages? Do you install them as well? Yes, these are the bioconductor packages. These ones here, the seq tools, the R subread, EDA seq. So we are installing everything right from up here, starting with the very first line, the Bioc Manager. If you don't have it, then install this bioconductor package, seq tools, R, subread, EDA seq, Lima, HR. When you're done with these, then you come down to the CRAN packages as well, run each line, install PV-class, and install the pHeatmap package as well. All right, so your path is not writable for which one? Did you run these installations at once, or are you running one by one? Which one in particular is not updating? So which one in particular? Is HR. Can you install the CRAN packages as well? And we see if you are successful. Install the CRAN packages. Meanwhile, for the rest of us, okay. Meanwhile, for the rest of us, type yes if you have finished installing the bioconductor packages as well as the CRAN packages. Okay. All right. So okay. Continue giving feedback on the chat. Okay, depending on how fast your internet is, these should be running. There are some colleagues who have been locked out of the webinar, removed by the host. Oh, um, let's see. So how do I know if successfully installed? If you successfully installed your packages, at each stage, you should have a statement that says the downloaded binary packages are in, the file path that will be given down here. Now, also, you will see these statements, the packages successfully unpacked, and so they are installed. And also, when we go to the next script later, when we are loading libraries, you should be able to see the functions. They should work. If they're not working at that point, it means you didn't install. But as you see them down here, you should check and know that you have successfully downloaded the packages. If there's an error term, it probably means that it was not successful. If there's no error term and you have the success message, then you should know that they have been successfully installed. They should be successfully installed. So please continue running this. Continue running this. Okay, continue running this. And then we can start the presentation. We can start the presentation. If they prompt you to update all sum, type A in the console down here. Type A in the console down here. Somewhere down here, you type A and then press enter. Type A and then enter. Small a and then enter. Oh, no. The type A is for when you have a prompt down here to do the updates. Okay, so if you're still running, please continue running. If you're still running, please continue running. Continue having those packages getting installed. We are now going to start the PowerPoint presentation. So I'm going to stop sharing my screen and hand over to Stephen.
Thank you very much, Peace, and thank you very much, everyone, for installing the packages. Those who are still installing, please keep going. So let me share my screen here. Type 1 if you can see the screen, the slides. Okay, thank you. So today's our fourth day. The first day we covered sequence analysis. So we started with DNA from the gene, and then how it is transcribed. Then you get your protein sequence. Then from there we went and did translation, and then after that we said that after getting your sequence, assume you're an experimenter, what do you do? So we started with the similarity search. One of the most commonly used method is called BLAST. It's a basic alignment search tool. There we discuss different types, but there are two types that are most commonly used. We have protein BLAST, BLAST P, that when you have your protein sequenced in amino acids format, it uses the database has the protein sequence. And then when you have your DNA, you use BLAST N, and your query is a DNA, and then the database is also DNA. Then after that, we looked at secondary structure prediction, because from primary, the amino acids are the primary sequence, and then look at secondary structure. We talked about the advantages of secondary structure. Then we also discussed the tertiary structure, which is 3D, because most of the proteins, basically the function is through 3D. Then we also discussed multiple alignments and alignment and why do you have alignment. So we mentioned the concept domain and so forth. And then that was the base of our next topic, the next three topics. So after that, we did metagenomics. So metagenomics basically is the study of genetic materials in an environment, and the environment can be soil, forest, gut, human skin, and so forth. And we went through the process of how you get your sample, you send it for sequencing. Then after that, you get it back, you do some cleaning, and then after doing the cleaning, you go through and line those sequences in databases to find out what type of microbes you have. We also saw that there are two types of metagenomics. There's targeted or amplicon, where you use markers, and we saw three markers: 16S RNA, that's mostly using bacteria and archaea, and then we discussed 18S, that's mainly using eukaryotes, and then ITS, it's using many fungi. Then we also discussed shotgun, that you do the whole sequence. And then after that, after sequencing, then what do you do? We discuss preliminary analysis that you do. You plot your data. We discuss the counts, and then how you convert those counts to relative abundance, and then went to statistics. We discuss alpha diversity, that is, you study diversity within a sample. Then we talk about beta diversity, that's diversity among samples, discuss different methods from invariant statistics to the rest. So that was about metagenomics, how you can use metagenomics. And we also discussed the application from agriculture to human to medicine and so forth. And it's being and people being using metagenomics. So I also showed you the tools that can be used in metagenomics. And those tools do the same things. You can do online tools. We had Mother, Chime, and the rest, but we are using R Studio because it's open source and free. And then because some of these tools you have to run them on high performance computer, or you have to run them on servers on the cloud, and sometimes you need a very good internet. So ARA would be a good way to go. So our next topic is transcriptomics, which is RNA-seq. So that's what we're going to spend at least today and maybe some part of tomorrow. If we don't finish today, we'll finish tomorrow. And tomorrow we'll finish the webinar with genomics. So what's RNA? What's RNA-seq, which is ribonucleic acid sequencing? Okay. So before we go there, I would like us to go back and watch this video that we watched when we on Monday, when we started from DNA to protein. But I'm not going to finish. I'll stop in the middle. And after watching that video, we started from DNA, we came DNA, messenger RNA, and then protein. So we're going to watch this, and then I would like to pay attention to what's going on from nucleus up to cytoplasm. And then we're going to watch another video again that explains what RNA-seq is. Because after doing that, then we're going to help us to understand why we study RNA-seq, okay? So let me go here and let's watch this. I will stop in the middle, okay?
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm.
Okay. So basically, gene expression deals with the messenger RNA. So I'm just repeating this. So either plant or animal, you have this chromosome, and these chromosomes are basically made of DNA packed around the histone, okay? So nothing happens if a gene is not switched on. But if a gene is switched on, this double-stranded DNA is going to, the enzyme is going to come. And this enzyme here, RNA polymerase, will come. It's an enzyme, and then it opens up the DNA. Okay. So these are floating bases just in the nucleus. But this messenger RNA is not going to be random, because that gene is going to be is being switched on because of a certain maybe environmental factor. And I'll go back again to maize, maybe because of the heat, there's no rain. So the corn plant wants to produce an enzyme or protein that's going to protect that maize from getting burnt. To do that, a gene is going to be switched on in the nucleus, and that gene should be transferred out so that it can be made, the protein can be made. But to do that, somebody has to do it, and that's messenger RNA. So it keeps adding this basis here. Okay. So when it's done, this messenger RNA is exported out of the cells. And when we watch this video, that was only one cell. So we have billions of cells of humans and plants. So their cells are going to be making those messenger RNA. So the study of RNA-seq is basically to quantify, to tell you that if you have this experiment, maybe treatment and control, which genes are going to be expressed more and which genes are going to be expressed less. And how are we going to find that? Because of this messenger RNA. That's why it's called RNA-seq. Okay. So basically, that's what gene expression, RNA-seq analysis is. You're trying to find out how the genes are being expressed and what type of, what's the quantity of that. Okay. Okay. So basically, I want to start with that. But now we're going to watch a video of the process of analyzing RNA-seq. Okay.
How long before I see results with Tai Chi?
You have to.
Day 3, you'll feel your energy. Day 7,
your
eyes will change. Day 12.
There's going to be some advertisement.
Even when you're feeling bad,
those advertisements, okay?
even when you've got the flu, even when you're down and sad, you can watch StatQuest. Hello, and welcome to StatQuest. StatQuest is brought to you by the friendly folks in the genetics department at the University of North Carolina at Chapel Hill. Today, we're going to do an introduction to RNA-seq. We'll start out with a bunch of normal neural cells and a bunch of mutated neural cells. These guys are abnormal. The mutated cells behave differently than the normal cells. We want to know what genetic mechanism is causing the difference. This means we want to look at differences in gene expression. So let's figure out a way to do this. Each cell has a bunch of chromosomes, and each chromosome has a bunch of genes, and some of the genes are active. These wavy lines represent messenger RNA transcripts. But this gene is not active. High-throughput sequencing tells us which genes are active and how much they are transcribed. This is super important, so I'm going to say it again. High-throughput sequencing tells us which genes are active and how much they are transcribed. We can use RNA-seq to measure gene expression in normal cells, and then use it to measure gene expression in mutated cells. Then we can compare the two cell types and figure out what's different in the mutated cells. For gene 1, there's no difference between normal and mutated cells. For gene 2, we see a big difference between normal and mutated cells. And for gene 3, we see a subtle difference between normal and mutated cells. There are 3 main steps for RNA-seq. 1, prepare a sequencing library. 2, sequence. And 3, data analysis. Let's start with preparing an RNA-seq library. Note, I'm using the Illumina protocol and sequencer as my example because they are commonly used. But keep in mind, there are other protocols and sequencers that do it differently. Step 1, isolate the RNA. Step 2, break the RNA into small fragments. We do this because RNA transcripts can be thousands of bases long, but the sequencing machine can only sequence short 200 to 300 base pair fragments. Step 3, convert the RNA fragments into double-stranded DNA. Double-stranded DNA is more stable than RNA and can be easily amplified and modified. This leads us to the next step. Step 4, add sequencing adapters. The adapters do two things. One, they allow the sequencing machine to recognize the fragments. Two, they allow you to sequence different samples at the same time, since different samples can use different adapters. This helps you save time and money. Notice that this step doesn't work 100% of the time. There are a couple of DNA fragments that didn't get sequencing adapters. In step 5, we PCR amplify the library. Only the fragments with sequencing adapters are amplified. They are enriched. Step 6 is quality control. You verify the library concentration, and you also verify the library fragment lengths to make sure they're not too long or too short. Hooray! Now we sequence the library. Let's see how this is done. Imagine this is a fragment of DNA we want to sequence. It's vertical because that's how it is inside the sequencer. Actually, there are about 400 million fragments laid out vertically in a grid. I'm just showing you 4 fragments so your brain doesn't explode. This grid is called a flow cell. The machine has fluorescent probes that are color-coded according to the type of nucleotide they can bind to. The probes are attached to the first base in each sequence. Once the probes have attached, the machine takes a picture of the flow cell from above that looks like this. The picture tells the machine that the first base in the bottom left-hand corner is an A. This base is a G, and these two bases are C. Then the machine washes the color off the probes. Then the probes are bound to the next base in each fragment. The machine takes a picture from above, and now it knows that this base is C. This base is G, and these two bases are T. Then the machine washes the color off the probes, and the process repeats until the machine has determined each sequence of nucleotides. This is how it works with 4 DNA fragments. With 400 million DNA fragments, the matrix is much denser. This matrix still isn't 400 million DNA fragments, but it illustrates one type of problem that can occur. Sometimes a probe will not shine as bright as it should, and the machine isn't super confident that it is calling the correct color. Quality scores that are part of the output reflect how confident the machine is that it correctly called a base. In this case, the faded dot would get a low quality score.
My boss thought I worked all weekend, but I just stopped using ChatGPT like a toy and do this instead.
Another reason you might get a low quality score is when there are lots of probes the same color in the same region. This is called low diversity, and the overabundance of a single color can make it hard to identify the individual sequences. The colors will blur together. Low diversity is especially a problem when the first few nucleotides are sequenced, because that is when the machine determines where the DNA fragments are located on the grid. Now that we've seen how the machine works, let's take a look at the raw data. Each sequencing read consists of 4 lines of data. The first line, which always starts with an @ symbol, is a unique ID for the sequence that follows. The second line contains the bases called for the sequenced fragment. The third line is always a plus character. I have no idea why. I asked the internet, and I don't think it knows either. The fourth line contains quality scores for each base in the sequenced fragment. A typical sequence run with 400 million reads will generate a file containing 1.6 billion lines of data. Now that we understand the raw data and how it's generated, we need to filter out garbage reads, align the high-quality reads to a genome, and count the number of reads per gene. So let's talk about how to filter out garbage reads. Garbage reads are 1. Reads with low-quality base calls, and 2. Reads that are clearly artifacts of the chemistry. We've already talked about low-quality base calls, so now let's talk about artifacts from the chemistry. A typical read is a DNA fragment plus adapter sequences. But sometimes the adapters just bind to each other and the read is just adapter sequence. This is a garbage read. Now we need to align the reads to a genome. We start with the genome and the genome sequence. We then split it into small fragments for reasons that will be explained in a little bit. We then create an index of all the fragments and their locations within the genome. Now we have a sequenced read. Just like with the genome, we split the read into fragments. We then match the read fragments to the genome fragments. The genome fragments that matched the read fragments will determine a location, chromosome, and position in the genome. Why are we breaking the sequences up into small fragments? This allows us to align reads even if they are not exact matches to the reference genome. Imagine this base wasn't in the reference genome because, for example, my genome is slightly different from yours. Then this fragment won't match anything in the index, but the other fragments will, and we will still be able to figure out where the read came from. Next, we have to count the reads per gene. Once we know the chromosome and position for a read, we can see if it falls within the coordinates of a gene or some other interesting feature. Here are 2 genes and their coordinates within the genome. We have this information for all 20,000 genes in the genome. After you count the reads per gene, you end up with a matrix of numbers like this. The first column contains gene names. The human genome has about 20,000 genes, so this matrix has about 20,000 rows. We're just looking at the first few. The remaining columns contain counts for each sample you sequenced. There are usually between 600 and 800 plus samples. Bulk RNA sequencing, where a sample is the average of a pool of cells, usually 6 million cells, might have 3 normal samples and 3 disease state samples, or a total of 6. Bulk RNA sequencing is the more common method used these days. It was the original method. Single-cell RNA-seq treats each cell like an individual sample, so it can generate a lot of samples. Each row gives counts per sample for a specific gene. If this were a single-cell RNA-seq experiment, we would have 20,000 rows, genes, by 800 plus columns, samples, giving us at least 16 million values to keep track of.
Do you understand my English but cannot speak? You know the words? Understand movies, YouTube, podcasts?
That's a huge matrix, and it's only going to get bigger since sequencing gets cheaper and people are doing more and more samples. The last thing we do before analysis is normalize the data. This is because each sample will have a different number of reads assigned to it due to the fact that one sample might have more low-quality reads, or another sample might have a slightly higher concentration on the flow cell. Here's an example. Sample 1 has a total of 635 reads assigned to it. Sample 2 has 1,270 reads assigned to it, twice as many reads as sample number 1. This does not mean that the genes in sample number 2 were all transcribed twice as much as in sample number 1. Instead, it means that sample number 2 had fewer low-quality reads and might have landed on more spots on the flow cell than sample number 1. However, the read counts make it look like the genes in sample number 2 were transcribed twice as much as in sample number 1. So we need to adjust the read counts per gene to reflect the differences in how many reads were assigned to each sample. The simplest method is to just divide the read counts per gene by the total mapped to each sample. However, there are many more sophisticated ways to do this. For more details, check out the videos in my high-throughput sequencing playlist. We started out with a bunch of normal neural cells and a bunch of mutated neural cells. Then we extracted the messenger RNA. Then we sequenced, aligned, counted the reads per gene in each sample, and normalized. Now it's time to analyze the data. Step 1 in any analysis is always the same. Plot the data. Remember, the data is a huge matrix. If there were only 2 genes, then plotting the data would be easy. First, we'd replace the gene names with X and Y. And then just plot the samples on an XY graph. Sample number 1 would go at X equals 30 and Y equals 24. Sample number 2 would go at X equals 5 and Y equals 10. And sample number 3 would go at X equals 13 and Y equals 18. But we have 20,000 genes. So we would need a graph with 20,000 axes to plot the raw data. So we use PCA, principal component analysis, or something like it to plot this data. PCA reduces the number of axes you need to display the important aspects of the data. This is a PCA plot from a real RNA-seq experiment done on neural cells. The WT samples are normal. The KO samples are samples that were mutated by the researchers. The KO samples make a nice little cluster in the corner. The WT samples are all on the left side, but spread out on the y-axis. The way these graphs are drawn, the most important differences are on the x-axis. Differences along the y-axis are not as important. This means that the biggest differences are between the WT samples and the KO samples. However, when we do further analyses, we may wish to exclude WT2. Excessive self-promotion. If you want to learn more about how PCA does what it does, check out StatQuest, Principal Component Analysis, PCA, Clearly Explained. This is a single-cell RNA-seq PCA plot from neural cells. The colors were added based on what we know about the cells. The green cells were stationary, and the orange cells moved around the petri dish. Most of the orange cells are separated from the green cells. However, there are a few orange cells that seem more like the green cells. If we want to determine what is different between these cells and these cells, we might exclude these cells from the analysis. In summary, plotting the data, 1, tells us if we can expect to find interesting differences, and 2, tells us if we should exclude some samples from any downstream analysis. Now we're ready for step 2 in our analysis. Identify differentially expressed genes between the normal and mutant samples. This is typically done using R, a programming language, with either EdgeR or DESeq2, and the results are generally displayed using this sort of graph. A red dot is a gene that is different between normal and mutant samples. Black dots are genes that are the same. The x-axis tells you how much each gene is transcribed. CPM stands for counts per million and is a type of normalization. Genes on the left side are lowly transcribed, and genes on the right side are highly transcribed. The y-axis tells you how big the relative difference is between normal and mutant. Log FC equals the log of the fold change between the two sample types. We've identified interesting genes. Now what? If you know what you're looking for, you can see if the experiment validated your hypothesis. If you don't know what you're looking for, you can see if certain pathways are enriched in either the normal or mutant gene sets. And then what? Check out the other videos on StatQuest. You'll find complete tutorials on all kinds of stuff related.
Okay, so basically, that's what we're going to cover. We're going to cover from how to clean the data, what are involved in that. So I wanted us to watch this video. Let's go back now to our slides. So if you can see slides type 1. Okay, thank you. So what's RNA-seq? It sequences the RNA present in a sample. It can be from a tissue, it can be from cells, it can be from organism. So by doing that, you determine the quantity of and types of RNA transcripts that are being expressed at a given time. So why does it matter? It provides a snapshot of transcriptome, what's being transcribed. So I still go back to the maze. So if you take the samples at that particular time, you give it a snapshot of which genes are being turned on and which genes are being turned off. Okay? It reveals how strongly each gene is expressed. Okay, that's what RNA-seq is. So, like in the video, you could see that these are chromosomes, and they had basically, it gave an example of 3 genes. These genes are not being transcribed at all. The way we are the messenger RNA. This one has only like 3, and this one has more. So this tells you that that factor that is switching these genes on, this one gives more what messenger RNA. Okay. So he gave an example between the normal neural cells and the mutated one. So for gene 1, because sometimes we have what we call housekeeping genes, they have to be expressed all the time. So whether you are normal or you are sick, these genes have to be expressed. So the gene on the left here, I think there are 3. It's expressing 3 messenger RNA, and the gene on the right also 3. So this is, they always call them housekeeping genes. But because this is mutated, basically something is missing. The normal gene at this particular time is not being expressed at all. And the mutated one, you have this one is not being expressed. So this, now if you do your RNA-seq analysis, this will tell you maybe that's why, maybe if this is a cancer cell, that's why this is cancerous, because of this type of gene. So something's happening here. Okay. Okay. So this is how we're going to do our analysis. RNA-seq. So in all RNA-seq experiment, you start with your library prep, the way you explained in the video. Then you send to sequencing. And they send you back the FASTQ files. And in the FASTQ files, you do your quality control. You clean your FASTQ files, and you get the clean files. The first thing that you do, you align your FASTQ files to reference genome, and they're always in a FASTA file format. So remember, when Peach told you to download those 2 files from Service, one ended with FA. That means that was a reference genome. So I will talk more about that data, but that data came from the yeast. So the FA is a genome, is a yeast genome. And then you align your reads to that genome. Okay, like when we watch in the video. But those reads, we know their position, but now you have to annotate them. You have to find out what these genes are. So with that, we do feature assignment, and that it's another file called reference annotation, because every organism, after being sequenced, they have the genes are always annotated. So these files are either GTF or GFF files. So for us, when the file that downloaded, it was .gff2, okay? And then after that, you get your metrics, the one he showed us. You get your metrics, and then after the metrics, you normalize, and you do the expression analysis. The rest, now you can go and do the downstream analysis. So we're going to go through all this process, and the R script that you downloaded are involved with all this, and we're going to work with you together. Okay, so we're going to start from this, where we got the sequence up to when we are going to get the functional analysis, okay? So I'm going to show you that how do you get your sequences from companies? As I mentioned yesterday, I have collaborators in developing countries. Most of the time we send them this here, the matrix. All this process is done abroad. But this is, if you send your sample, always a company, you open an account, they give you a username and a password that is only known maybe to you to protect your data. So when everything is sequenced, you log in, and they always keep your data for 6 months. And this is how you get it. You see all this data on the web page. You have to have at least very good internet. So instead of, and these are heavy files, by the way. So the best way to get these files is to do batch, get them through batch. Batch, what I mean, just you use like FTP tools like FileZilla. So they'll give you a link that will use your FTPs like FileZilla or other FTP tools. But sometimes you can, if you're using command line, whether you have either Mac or you have Linux, you can just use the command line and get your files, okay? So I wanted to share that with you so that in case you are working with a company, one is your data is mainly being kept for only 6 months because they have to, it's a business. And then how do you get the data? They assume you know, they just send you the password. But the easiest way is to use FTP, a batch way. Otherwise, it might take you a long time, and if you have a very slow sequence internet, okay? So after downloading, this is basically what you get on your computer. So each file comes like in a zip, and then becomes a folder. And then within a folder, always you have the forward read and reverse read. The forward read is always represented by 1, reverse read by 2, and almost all these files are zipped because they're big files. And then the MD5 text always tell you about the sequence. And don't worry about these ones here, but always you have -1 is a forward read, and -2 is your reverse read. Okay, and by the way, these are zip, but look at this size. 1.65 GB, 1.7 GB. These are huge files, by the way. Okay. So why do we study RNA-seq? Because RNA-seq, like metagenomics, has application in agriculture. It helps in crop improvement. You can identify stress response genes, maybe in drought, in heat and salinity, also with pest and disease resistance. So sometimes you can find a susceptible or resistant plant, and you find that the genes that are making that one resistant is through what? Through gene expression analysis. Tomorrow we'll talk about genomics, and that will help you find the particular marker. But you start with RNA-seq. Also help with determining yield and quality. So with that, you can uncover the pathways. So that is, we're going to see that in a downstream analysis. We're going to use KEGG, we're going to use gene ontology. Also in breeding programs, it helps in marker discovery and for genomic selection. And then in livestock, it helps in studying immune response and so forth. So this is in agriculture. And then in health, you can compare disease and healthy tissue. Like the video was basically that was in health field. You can also help them in biomarker discovery, pathway analysis. You can see which pathway is being disrupted and which pathway is it's causing cancer and so forth. Another area that helps is mutation and variation detection. So if you have different variants, it's going to help you. So that will be even the first process of how you're going to start your SNP analysis. It also can help with fusion transcript, like in leukemia and so forth. So that's why we study RNA-seq. So I'm not going to spend much time here because we covered this in metagenomics. Also, he talked about this in the video. So this is basically the format of FASTQ files. So when you're doing metagenomics, RNA-seq, and genomics, always you're going to have this type of files. And I'm not so going to spend time here. We talked about this yesterday, and we always looking for first score of 30. That means that the error of the machine is 1 in 1,000. So if the machines sequence the DNA, at least you can live in 1 in thousands, okay? Okay, so we're going to start with quality control. So when you get your sequence, the first thing you have to do is quality control. So that's what we're going to do in RStudio. But before we go to RStudio, I'm going to share my screen and discuss this data here that we're going to use. So this data came from yeast experiment. So we have 3 treatments, treatment 1, 2, and 3, and each treatment has 2 replications, and we have 3 control. Control means wild type. So each treatment was sequenced forward read and reverse read, forward read and reverse read, okay? So that was the data set. Okay, and then let me go back. If I can share my, if I can share my drive or my, let's see here. Okay. Do you see my folder, RNA-seq folder? Type 1? Okay, thank you. So you downloaded this sequence, these are the sequences. Then you also downloaded these 2 files. So this is an annotation file that's going to, after aligning, this has all the information about the genes in yeast. And this one is the genome of yeast. So this is the one that we are going to align our sequences on, okay? So you have the annotation file, which is has GFF3, and you have the genome, which is the FASTA file. FASTA file ends with FA, okay? Okay, so before we go to our RStudio, I would like also to share how we are going to do the analysis, okay? So we already watched the video. You get your sequence, you do the quality control and so forth. So the first thing that you guys did was you install the package. Okay, went through that with peace. The second thing we're going to do, we're going to open our RStudio. We're going to check the quality of our RNA-seq. After checking the quality of the RNA-seq, we'll clean. For example, if the Fred score, all the sequences with Fred score that are lower than 30 will be removed. And then the frequency of each DNA, each base, A, T, C, and G, the ones that are not very good, we remove them. And then after that, we'll check, because we're going to be visualizing them. Then we'll check the ones that have been cleaned, because after cleaning, the clean reads will be put in a different folder. After getting our clean reads, then we'll do the alignment. We'll use the RR script called RNA-seq alignment. So when we're doing alignment, we are aligning those reads to the yeast genome. Then after aligning, we'll now annotate, give them their names. And then after that, remember, the first thing that that gentleman said was to do data exploration. For us, we're going to visualize that one. We're going to use PCA and a principal coordinate analysis. Then after that, we're going to filter and normalize, do normalization. And then we're going to do differential gene expression, and then we'll do the downstream analysis. So this is the process of doing RNA-seq. The way he talked in the video is what we are going to do. Okay, so this is the process. And you have all these scripts that you downloaded. You already done this. Next, we're going to go through and so forth. Okay. So we'll be going between the scripts and the slide. After we run our script, and then we explain through using the slide. Okay. Okay. Go back to our slide here. So we're going to do the first quality control and trimming. Why are we doing that? You already explained that in the video. We're going to remove the adapter sequences. You're going to remove poor quality bases at the start, at the end of the read, because we're going to remove the masking and so forth, okay? So to do that, one of the package that you download is called sectors. So this provides function of analyzing nucleotides. It's also detect GC content and also detect the Fred abundance, okay? So what we're going to do, I would like you to go and open your RStudio, and I'm going to share my screen and you follow me, okay? So if you can see my RStudio type 1, open, I'll wait for you. Let's see your RStudio type 1, okay. Let's see my RStudio type 1, thank you. I would like you to go to your RStudio. You're going to bring those yeast sequences in FASTQ format. We're going to check them. And then after checking them, we'll clean them, okay? So we're going to start with this here, okay? So what we're going to do, you follow me, we'll make sure that you are in your RNA-seq folder. So right now, make sure I'm in the right folder here. Right now, let me see here. Right now, you should be in your bioinformatics folder, I think, because each time you open, you should be in your bioinformatics folder. But remember, all the sequences in the script, when you downloaded, you put those in RNA-seq folder. So we have to make sure that we are working from that folder. Okay? So We can do that in two ways, but let me try this one here. If it works, then I'll tell you to do. If not, then I'll tell you what to do. So I'm going to first open the file in this RNA-seq called the file here. Quality check, RNA-seq check. Let me go back here to make sure. This one here, you see this file here? Open this file, RNA-seq quality check. You should be having that on your in your folder. If you have opened that, type 1, it should look like this. Okay, so yours, it doesn't have this. I checked this, but yours does not. So this water, can you try to run this and see this line 3? If you run that, see what happens. If you don't get any error, you run line 3, and if you don't get any error, type 1. If you get an error, then we're going to go and do it another way. If you get an error, if you don't get an error, type 1. If you run line 3, somebody typed 0, I think they have an error. Or somebody got an error. So those ones who were successful is okay. Those ones who are not, there's a way we're going to do it. Okay. So there are two ways. You could run this script, and that's okay. But if you got an error, follow me. Follow my cursor here. Okay. You see these 3 dots. These 3 dots here. See those 3 dots? You click on those 3 dots and go to your bioinformatics folder and click on that RNA-seq and open it. Okay, I'm going to do it again. You follow me. See these 3 dots here? You click on those 3 dots, go to your bioinformatics folder, click on RNA-seq and open it. So if you've done that, like type 1, those ones who did not succeed, if you succeeded, don't do that. If you ran this line 3 and succeeded, you don't have to do what I'm doing. So if you've opened it, okay. So now we have another extra step that we have to do. Okay? Okay, so this is now the last step that we have to do. You see this kind of like a small round ball here in green, green or blue in color. And on the right is more. If you click on that, you'll see all these kind of like a window. So you click on set as a working directory. If you click on that, it will set you as a working, it will set your RNA as a working directory. Okay, I'm going to repeat. This one here looks like a small wheel here. On the right, there's a more. If you click on it, you come down, you click on set as working directory. If you click on that, then it will take you will be in now RNA-seq working directory. Okay, let's start again. For those who line 3 didn't work, for those who ran line 3 for the RNA-seq underscore quality check, you don't have to do it. But those who got errors, this is how we're going to get into RNA-seq to make it our working directory. The first thing you have to do, there are 3 dots here. You click on those dots. You come here. You go to your bioinformatics. You click on your RNA-seq, and you open. That's the first step. And the second step, on the lower right, you have this kind of like a small wheel here, blue in color, light blue. You click on more and set it as a working directory. So if we are all if all of us are now in RNA-seq, you should see like home, desktop, bioinformatics, and RNA-seq. If you look at where it looks like a house, there's a home, there's a desktop, there's bioinformatics, and RNA-seq. If you see that type one. Okay, now we can start doing our analysis. So the first thing we're going to do, we're going to call those libraries that we installed, okay? So we're going to run line 6 and 7, basically to call the libraries. If you didn't install those libraries, you're going to get errors. And then we're going to have our path where the RNA-seq is. And then you're going to run up to 16 and see if you don't get any error and let me know. Up to 16. Somebody's got error. What does the error say? Okay, the people, if you're getting an error, that means you were not successful in installing your what? Your package. Remember, Peace told you to go install one by one. That means the package wasn't installed. So if you were not able, if you didn't install the package, then this won't run. So go back and install your package. So if you're getting all this error, you have to go to the RNA-seq working directory using what we have done here. Error unexpected this, error unexpected. Why somebody have this greater than sign? It should not be there. Okay, so if you're getting that there's no package called RFSU, that means you did not install your package. I mean, the first thing that you did with Peace was to install those packages one by one. Okay, so it's still running, it's okay. Okay. So okay, those who are successful up to 16, can you type 1? Okay, how to install the package again? Okay, so let me go back and because it seems I think all of us, majority of didn't install the package. So I'm going to stop here. I'm going to go back and we're going to install the package, okay? So you follow me here. Go to File, Open. In your RNA-seq folder, there is a file called RNA-seq package. Let's see. There's a file, this file here. RNA-seq package. Let's see, where is it? Somebody help me. RNA-seq package.
It's down.
It's down?
It's down.
Oh,
Yeah.
this one. Thank you, Peace. So this is the package. It seems like big percent of us didn't install this package. So we're going to do this slowly, line by line, because without these packages, we are not going to do anything. So I'm going to install this line by line. Okay, start from line 1, you run. So don't just do it all at a time. Okay, you see this? Update, you look at the console. Update always you type here small a and enter. And let it run. Let it run. So we're installing sec tools. Let it run until it's done. Okay, you see when it's done, here it tells you the downloaded file binary package is in this directory. Okay, when that's done, don't worry about the what? The warnings. We go now to R subread. We're going to run that. Again, it's going to ask me if I want to update all. I'm going to type A. Okay. Because without this, we were not going to do anything. So this is done again. So the downloaded binary package and is here. Now I'm going to go to EDSec. I'm going to run that. Again, it's going to ask me if I want to update all. I'm going to say yes. Enter. Okay, that's done. The next, I'm going to go to Lyman. Okay. Again, A. So these packages from 4 to 8, these were Bioconductor. So those are installed. Now I have to install also from CRAN and have two packages from CRAN. I have PV class and PH heatmap. I have to install that. Okay. Just, if I say yes, gonna disorganize my because I already installed that. Nope. Okay. So it's also going to ask you because we already installed this. I said no. So unless you have this, those scripts will not work because they need these libraries. It's like when we did QGIS, when we're doing data science and data management, we had to install those modules. See that? Okay, so those who didn't install this in the beginning, type 1 if you're now successful, so that we can go back to our analysis. I'm gonna give you at least 5 minutes. Okay? Don't worry about the warning messages. Okay. If you see the update all, just type A, like exactly what I was doing. Right now, don't go to setting directory first. Let's first finish the installation of the packages. We'll come there. Okay. Someone say you were too fast, sir. Okay, let me get back again. Okay. So before we do anything else, we have to install these packages. And these packages are found in this script, RNA dash sec dash package. If you have that, open it. After opening it, start running from the first line. Okay. If you run, mine already installed, but it will ask you if you want to do what? To update, and you type a small a. Okay. Okay. So this was my second one. When you click on the line, it will ask you update all, some or none. You say update all. How do you do that? By typing a small a in your console and you enter. Okay? And then it will install. Then after installing, it will give you this message here. The downloaded binary packages are in this directory, depends on your computer. Okay? And then you go to the next one. You do the same. Again, you type A. Okay? And you come to the last one for bioconductor. You type A. Okay? So when you're done with bioconductor, You come to cron. You click on this, you run. Me, I already have this one installed. I'm going to say, I'm not going to install it. I'm just going to cancel that. But for you, you have to install it. Okay? Because if I have to restart my computer, it's going to disorganize what I already have here. Okay? You do the same here. You don't cancel. But me, I'm canceling because I don't want to restart. It's going to disorganize what I already have here. That's what you have to do, really. Okay? So I'm going to give you at least 2 minutes. Then we're going to go back to our quality control. Okay, so let's start analyzing our data now. So you need to go to your RNA-seq folder. You need to bring this folder here. Look at my RStudio. I'm going to start file, open. I'm going to use this file called quality control. Quality control here. Let me see, where is it? Quality check. This one here. RNA-seq quality check. I would like you to open this file. Okay. Before we went back to reinstall packages, we did two things. One, now we want to be in our RNA-seq directory. The one way for doing it was to run, we start with, was to run this, to set this as a working directory. But other people wasn't working, they were getting errors, but other people worked. I told to run line 3. If I do this, mine works. It depends on the computer sometimes. But for those who did not work, we went through another route for you to be in RNA-seq folder directory. To do that, you go to these 3 dots, okay? And then you go and open RNA-seq. After opening, there's this wheel here. On the right, there's more. If you click on that, there's set as a working directory. If you click on that, it will set you, me, I'm already there. In a RNA-seq becomes your working directory. How do you know it's your working directory? If you come now here where there's home, this house here, then you see a desktop, you see Bioinformatics, because this is where you were before. But now, RNA-seq is the last folder. When you started your computer, Bioinformatics was the last folder. But now RNA-seq is your last folder. Then that's where now your directory where you're going to work. Because all your data that you downloaded from the Google Drive are stored here. Now, if you're there now, we're going to start analyzing our data. The first thing we have to do is to bring our data sets, the treatment and the control. We are going to import them to our RStudio. So that is done from line 5 to line 16. So line 5, you're going to import them using this library here. And this is the pathway they are found. And this is, you run from line 6 to 16. And then I ask people who have been successful to put one. That's where we stop. So I would like it to run from line 5 to 16. And if you have run that, and in your environment, you see file names with 1 to 12, you have a path. And then on your console, you see the path of the files, and then the number of files, and then the reads, like what you're seeing on the screen, type 1. If you run this up to 16 and you see something similar to what you're seeing on the screen, you type 1. So for those who were getting error in typing directory, I would like you to go through the second route. I would like to go to the second route because you are not yet in RNS directory, by the way. That's why you're getting that error. You see, these ones here were getting the error. When you go to the second, okay, if you see that you have RNA-seq, you skip line 3. Okay, so this is what I would like all of us to do. All of us should do this. All of us should do this now. You go to line 3, all of us, and put a hash where it says set working directory. Put a hash before set working directory. Let's all do that. Let's all do that. Put a hash there and save it. I think that's going to help us. Let's all put a hash there. Okay, so we are all going to do this now, all of us. So let's just don't use this method at all. Let's all put a hash here so this doesn't work. Okay, so we are going to set our working directory using a longer route. To do that, you follow me here. You're going to click on these 3 dots. You're going to go to your Bioinformatics. You're going to go to RNA-6 and you're going to open that folder. And then after that, you look at this kind of like a wheel. You're going to go to more and set it as a working directory. If you do that, you will not be able to get any error. Because even if you start from line 1 now, this won't run. I think people are maybe accidentally running this. So we already got this hash here. This won't work. So if you can do that, then you won't have to worry about getting this error. Okay? So if you're done with that, run up to line 16. That means now we have our data already in Error Studio. Okay? So the next thing we're going to do is to do quality control. So you plot line 22. Just run line 22. You should get something like this. If you get something like this, let me know. And I will explain what we are looking at. Type 1. This error here. Okay, so these color codes here, we have 4 bases. You have A, C, G, and T. And in every organism, their proportions are different. Like in yeast, the proportion of A and T are higher, around 0.3 on average, and C and G are lower. But remember when that gentleman was talking on the video, he said in the beginning of the sequence, you get poor quality, and this is it. You can see how all this is just not consistent. So when we are doing our quality control, we have to remove this part here. You see that? Because it's going to affect our analysis. So from here onwards, okay, because the proportion of A and T in yeast is higher, 0.3, it's on average in the genome. And C and G are lower. This is perfect. But you can see if from the first read to around 15 is not consistent. So the next thing we have to do is to remove this part, all this from all the sequences. So that's the quality control he was talking about. And how did this come about? From sequencing, okay? From sequencing. So we have to remove this from what is called base calling, okay? The second, line 23, we are looking at the Fred score. I just leave 3. Yeah. So this is no, this is in 23 we are looking no, we are looking at sequence 3. So let me go here and plot the Fred score. You plot 29 and 30. So you look at 29 and 30. So our sequences, they're really, all of them are really good. They're above 30 Fred score. So we don't have to do anything with cleaning that, but we have to clean the first part of our sequences. All of them, we have to clean the first part of it. So that's his quality check. Okay. So if you run now from the top up to 29 and you don't get any error, type 1. So that was the way we're checking the quality. Then next we're going to go now to how do we clean this, the part that the guy called garbage. Let me go back and see what people are saying in the chat. It's in the chat. Okay. So somebody still failing to import, did every step right, said still failing to import. Yeah, still the setting working directory. Yeah, what I was telling you just after putting that hash, you should not be able to get this because, yeah, this is, I don't know why you're still getting this set working directory. Error unexpected. Yeah, this is a problem for HA. You're not supposed to get this because we already put a hash there to prevent it. Don't run if you, in case you didn't put hash, don't run line number 3. Start from line number 6. Try that. Try that one. Okay. Okay. So next we're going to do the cleaning on, and then this is a script called RNA-seq cleaning. If you can get that script. If you're able to get that script, type 1. RNA-seq cleaning. Okay, so what we're going to do, basically, is going to clean and put in another directory called clean underscore Q30. But I would like you just to run from line 5 up to just run the whole thing. What's doing is cleaning it, removing the bad part, and putting it in another directory. Try to run. If you don't get any error, let me know. So if you run that, I'm gonna go to the slides and explain what happened, and then I'll also share with you the script that will check again. Okay. Let me go back to the slide here. So this is what we did. This was the thread. So what we did was filter and trimming, garbage in, garbage out. So after running, it created a new folder called clean_30. And within that, the folder, we have the clean new FASTQ files. So if you go to your RNA-seq folder, you're going to see a new folder called clean_30. And if you open that folder, you'll see new sequences. Those are the clean ones now. So if you are successful in running that script for cleaning, you should be able to get a new folder. And that folder is called clean_30. And if you open that, you should be able to get those clean folders, those clean sequences. So let me go and open that and see if we have it. It's like it's this one here. And then it also gives a summary of the number of reads that were original and that were the ones that were out. So if you open this file, CSV file, you can see the first one had like 113,000, and after removing the batch sequences, the reads became smaller. Okay. Okay, I'm going to repeat again, and then I will ask question. Okay. So we went through and checked our reads, our sequence. We found that some of them were not good. But now we have to clean those ones. And we have a script for cleaning called RNA-seq_cleaning. So if you run the script, it's going to chop the first parts that the DNA, I mean, the bases were not, the proportion were not consistent. So chopping those, the first 15 sequences, I mean, 15 bases, is going to create a folder and put the clean sequences in the folder. Okay. So somebody type, if you have been successful of cleaning your reads, type 1 here. Okay. Type 1 have been successful in cleaning. Oh, people are still getting error. No row found in the desktop. So that means you did not put the when you work with PIS and you download the US sequence, you did not put those sequences in the RNA-seq folder. If you get this error here, no row files, that means you don't have those folders there. Okay. So the person who's getting this error here, you are not, I don't think you have your packages. I don't think you installed the packages for doing this function here. So go back and reinstall your package. Yeah, go back and restore your package. Okay. Let me go back now to the slides. Okay. So this script here, we're going to run to check the quality of the new clean sequence. We should get something like this. So the part that had all these wavy lines were all gone. Okay. And to do that, there's a script called RNA seq underscore quality check underscore clean. If you run that, you should be able to get some, you should be able to see something like this. Okay? So basically, these clean sequences are the ones that we'll be using for our downstream analysis. So this R code here, when you run it, it will check for you. If you go to that folder that we created and check to make sure that the part that we saw in the beginning are all gone. Okay? Okay, so let's go back to our studio. So what did we do? We loaded our sequences from RNA-seq folder. We checked for the quality. We found out that the FRET scores are good. So there's nothing to do with those. That's okay because all of them were above 30. But the bases in the beginning of the run of this when machine was sequencing, there was a problem because the proportions were not consistent. There were those wavies. So what we did was we ran this cleaning R script to remove those parts. And then what the script also did, create another folder for the clean sequences and put those there. So the next we are going to check the quality of those clean sequences. So to do that, there is another script call you open. It's called check clean, this one here. RNA-seq quality check clean. So we're going to check those one or two of those that have been clean. Okay. So if you open that and you open and run. So let me just go there and run up to 22. Okay. So you see this? So basically, this is representative of the clean sequence now. So now we are very confident that anything that we do with these ones, we've got what this guy was calling garbage. We've got all those are gone. Okay? So we didn't do the FRED because when we did the checking, our first scores were good. So up to this point, what we are doing is just cleaning our reads, quality control. We haven't done anything yet. We're just cleaning. So we got our sequences, we check them, and now we clean them. So what do we do next? We have to align them. Let me go back and check out some questions here. Yeah, this error set to S, the only error we're getting the error set directory. And then also the error, there are two errors that we've been getting. One is when people didn't install the package. And then two is the setting of directory. So if you can manage to get those, I think then we should be okay. Okay. So next, after cleaning your sequence, we now want to know where do these reads align on our genome? Where do they align on our genome? And that's where now we're going to use the yeast genome with the FASTA format. Where do they align? Okay, so that's where the alignment comes in. So the first 3 run that we did was quality control. Okay, the first sequence, the check was we brought our sequences in, we checked, we found problem with the first 15 first 15 rows because of the base calls were not very good. They were those wavy lines. And then our second script was to clean them. The third script was to check again if those wavy lines were still there, but we found that the wavy lines are gone. That was one, was quality control. Next, we go to alignment. How do you do alignment? You use these clean first file sequences, and then now you need the reference sequence. And our reference sequence is one of those two files you downloaded from the Google Drive. The one with an FA, and FA means a FASTA file. We discussed FASTA files 2 or 3 days ago, what a FASTA file looked like. So all genomes, most of all the genomes are in FASTA file format. So we have these reads. These reads came from sequencing. The sequencing came from your lab, somebody's lab, and that somebody's lab did experiment. There was a treatment and control with the yeast. And then when they did that, the genes were expressed, like we watched in the first video. So when the genes were expressed, messenger RNAs were made. And then they got the messenger RNA and then were converted to DNA, reverse transcript were converted to DNA, like we watched in the second video. And then they were sent for sequencing. And then the sequencing did their work. The sequencing company did their work. They sent back the FASTQ files. But these FASTQ files, of course, as we watch in the second videos, sometimes it's called calling. That, I mean, taking those pickers, that's what they call calling. So some of them are not good. That's why we use the Phred score. And then also the proportion of the bases were not correct. We did quality control. Now we have clean sequence. We're going to do the alignment. We're going to align this to the reference genome. We're in the second stage now. Okay. So how do you do the alignment? We do the alignment with this package called rsubread. So what it does, it has some function. One is called build index. Remember, when we watch the second video, that gentleman said you need to build index. Building index means you need to find exactly where in each genome you need to assign a coordinate. It also does this. So the first thing it's going to do is going to build an index. Because when you're doing alignment, this is what happens. Your research is just going to be searching all this genome, okay? So when you search all this genome and they say a match, how are you going to tell if what? If this read matches a certain part of the genome, you have to index it. You have to index it, okay? So this is what we watch in the video. Why do you do the indexing? Because sometimes you have different, people are different. So sometimes a certain part of the genome you might not have with the other one. But if you have those, it will tell you where the fragment is matching from. Okay? So this is what build index does in a layman's language. So this is yeast genomes. So the human genomes has approximately like 20,000 genes. Yeast has less of that. But each of these has its own names. So what this build index does, it goes and get information from all in the genome, and then it assigns a number like this, okay? So the first thing to do before you do the alignment, you have to build the index. And after building the index, basically you have to know where, if a gene aligns here, where is it coming from? Basically, you are trying to identify where they're coming from. That's why the build index does. And then next, you perform the alignment. We already seen the alignment, how to do the alignment a few days ago. So basically, the top is your reads, and the bottom is the reference genome. So where there's alignment, you can see, like here, it's just the same like we did, okay? So this is the cartoon. So this read one is aligned here. Another read is aligned here, another read aligned here. So this is now where it's very, very important where the index comes from. So let's go back here. You see this gene here is represented by this. This is the name of it. And this is the index, okay? So now when we do the alignment, you see this align to this index, and this is the length. This align the same, still the same, and so forth. So it helps us to know where these reads are being aligned, okay? So we're going to do that in RStudio, and there's a script for doing that called RNA-seq alignment. So what it does is going to create an alignment called, this is the name of the alignment. It's going to get the first read because you have forward and reverse. The type is RNA, and the file format is zip file. And then the output can either be SAM or BAM. BAM is basically a binary form of what? Of SAM, sequence alignment file. And the orientation is reverse and forward, and don't worry about the threading. If you had only one read, as I said last time, if you don't have enough money, you can only do one read. Then this will be only R or F, depends on which side. But we have two reads, so we have reverse and forward, and then after that, the alignment will be done. Okay. So I want to go through this before we do our RStudio. So let's go back to RStudio. Okay. Let's go back to RStudio. And what we're going to do, we're going to go and open our alignment. Okay. Let me check if I have some more questions here. Error lengths. So any error that says if a function does not exist, that means we don't have the package. Let's see here, error length. But I don't know why this error length is coming in here. Links is greater. So okay. So if you get error of length, that means you are not reading the file. That means the file is not coming, and the reason is you don't have the package for that. You left behind people with the working directory issue. Up to now, I haven't solved the problem. Well, okay. So people, okay. Let's do this. People who are running up to alignment, you run, let me go back to working directory issue here. Let me go back to working directory issue here, okay? Okay. So the people are going to, you run alignment and stop there. Let me go back and work with people who have been left behind. Okay, so what we're going to do, we're going to start from the beginning. The people who have run up to alignment, you don't clear your windows, okay? I'm going to start with people who have been having that problem of directory. I would like those ones to click files. And what I'm going to do is I'm going to start from 0. I'm going to go and be in my bioinformatics directory. Okay. So for those who have the problem, I would like you to do this with me. Do you see where there's home, a desktop, and bioinformatics? If you see the three, type 1 in the chat. If you have home, desktop and bioinformatics type 1. Anybody who can see that type 1? No, right now I'm working with people who have been, okay. If you can see that part type 1, yeah, if you can see this, if you can see home, desktop and bioinformatics, because these are the people who have been having problem. If you have this, then we are in the same page. Those ones who have an alignment, just wait for us. We're coming. Okay. Okay. So when you open your, assume when you open your studio this afternoon or morning or even where you are, you are supposed to be here. You're supposed to be home, desktop, and bioinformatics. If you're not there, then you didn't create your project. Because if you created a project, you should be there. Okay. So I think that's why we're having problem. So those ones who are not here, type 0. Those ones who are not people who are already in alignment just will come there. Don't type anything. I would like those who have been struggling and they cannot see this home, desktop, and bioinformatics to type for me 0. Okay, I think we still have a problem here. Okay, so this is what we're going to do. So this is what we're going to do. Because I think we are many here. So whatever you are, this is what I would like you to do. I would like us all to be in this directory now, all of us, those who are struggling with me. So I would like you to go, if you're not seeing this, I would like you to go to this, you see these 3 dots? I would like to click on these 3 dots and go and look for where your bioinformatics folder is. I would like you to do that and open it. Okay, I would like you, whenever you are, to go to these 3 dots and look for your bioinformatics folder and open it. So I would like all of us who are struggling to be up to here first. If you open it, let me know. If you open, let me know and type 1. Click on this, look for where your bioinformatics folder is and open it. If you've done that, type 1. Okay, I think we are getting there. Okay, so now, if you're all there, I would like you see this kind of like a wheel, kind of light blue. I would like you to go on more. If you click on more, you'll see this one, copy to. There's where it's called set as working directory. I would like you to click that. Okay, there's a wheel here, which kind of like light blue. There's more. I would like you to type set as working directory. If you click on that, now you should be able to see your home, desktop, and bioinformatics. If you can see that, type for me 1. Now you should be able to see this home, desktop, and bioinformatics. Okay, we are getting there. Good. Okay. Now, what we need now to go to our RNA-seq. Okay. We're going to do the same thing. We're going to go to these small 3 dots. We're going to go and click on your bioinformatics folder and click on your RNA-seq and open. Again, which is like what we did for bioinformatics. We're going to go click on that. We're going to click on our bioinformatics folder. We're going to see now our RNA-seq, and we're going to open, okay? And when we open that, we're going to do the same thing. We're going to go for that wheel. We're going to go to more. We're going to go to click as a working directory. Now, you are in now your RNA-seq directory. You should be able to see your home, desktop, bioinformatics, and RNA-seq. So in this now, you can run all the scripts that we have been running, because unless you don't have the package, because now you are in the correct directory, you can now start running those. So if you can now see this, type for me 111. If you can now see home, desktop, bioinformatics, RNA-seq, type for me 111. Okay, somebody still has 0. So I think we have pretty much brought almost everybody to where we're supposed to be. So since we were many left behind, I'm going to stop at alignment, and we're going to go back and start from cleaning, okay? So the alignment people will just wait for us, okay? Okay, so first thing we're going to do is we're going to load our data. We're going to load our data, and we're going to after that, we're going to see how the quality of our data. To do that, we're going to open there's an R script called RNA-seq quality check R. If you open that, okay, before you run it, I would like to do something for me. Please go to line 3 and put a hash before set working directory. Put a hash there before you do anything else. Do me that favor, and after that, let's save that, because that has been causing problem. What we went through, this line would have done it, but it depends on the computer. So after putting the hash now, you can now run this. Assume you installed your package. You should be able now to run all this. You should be able now to run all this. You should be able to run all this to see this graph here. And this graph, the way I explain it, is that in every organism, the ratio of A to T and C to G are different. So for yeast, A and T, the ratios are higher compared to C and G. But in the beginning, when the machine starts sequencing, doesn't do a very good job. So what we have to do is to remove this part, and that's what we did after that. That's right. So we also check the PRED score, and when we're checking our PRED score, remember I said PRED score should be at least 30. So our sequence, the PRED score are good, so we don't have to do anything with it. Okay. So this one was our first script, was just to load this sequence, there are 12, and check for the quality, okay? And then the second script that we used was the cleaning one. The cleaning script was to remove the first 15 nucleotides. That's what we did. Somebody still has a problem here. If you don't have anything to do with library, that means you did not install. So with that, you have to go and install, okay? So that was the first thing we did. And this was the name of the what? Of the R script. The second thing we did was we did the cleaning. So the cleaning, we use another script. And this script was called clean. And this is the script here. This script here. So after we ran this script, okay, we ran this script, and what this script does, it gets the sequence, it is chop up the first 15 nucleotides and put it in a cleaner folder. Okay, and that folder is called clean underscore Q30. And when you run that, okay, so we did that. After that, we checked for the clean sequences. If we got rid of the first 15 nucleotides that were not good. The other guy called them garbage. Okay, and that was quality check. Okay, so that script was this one here. That script was quality check clean. Okay, so when we run that script, we got this. So indeed, we removed the bad part. So then I went and explained alignment, because now we need to align these clean reads to our what? To our genome, which is the yeast genome. And you downloaded the yeast genome. So that's why we stopped, and we told these guys to wait for us there. So we're going to run the alignment and we pick up from there. Okay? So the script is called alignment script. So what we're doing, we are now aligning the clean script to our genome. And this is what's going on. You can see the alignment is taking place. Okay. So after alignment, okay, so the alignment people, we are back now. We can move up from here now, okay? So when you do the alignment, it creates another folder called a BAM folder, because a BAM is a binary format of SAM, which basically is an alignment file. So this is where now your alignment file is being what? Is being stored. It's called a BAM file. Okay. And if you open it, this is our aligned file here. The aligned file, they end up with the BAM format. If you go to your folder in RNA-seq and you are successful, you should get something like this. So this is now our aligned reads on the genome. Okay. But now we don't know, we have to annotate them. We have to know what part of genome are these aligned to. Okay. So this is bring us to what's called a future assignment, basically. So what's a future assignment? There was a file that we also downloaded. It's called GFF3. So as I mentioned, when an organism is sequenced, the genome is in a FASTA format, and then all the annotation becomes in a GFF format, okay? So let me see what's the next one here. Somebody has a question. Still the package issue here. So a package sec2 is not available for bioconductor version. Yeah, so you're getting this error because, again, you cannot install in bioconductors. So we have two major problems. It's either the file is not there or the thing was not installed. So this is now our number 3. So we did number 1, quality control. We did number 2, alignment. Then now we have to annotate. We need a file. And this is the file that you downloaded that had .ggf3. So after you're going to use the alignment file called BAM with this GFF. And then after that, you get a count matrix. Remember when the other gentleman showed us now that this is now on the left are the genes, on the top are the samples, and then in the middle, this is exactly what we're doing now. We're now going to find the counts of how many counts for every gene was aligned. This is number 3 here, okay? This is what we're going to do next. So we did one, we did a line. Now we are annotating. And after that, we're going to get our count matrix. It's like when we did metagenomics, we got also a matrix of the number of amplicon variants. Now this, we're going to get the number of reads, basically representing those RNA-seq that were expressed. So this is the part here. Okay, so we're going to go back to our other studio. There's a file for that called annotation file. So we open that, this file here, annotation. So if you look at this file, I'm going to go through with this file, with this here. So we're going to use these libraries here. I want to pay attention here. So here's going to get all the alignment from BAM files. See that? And then after that, it will get using the GFF file. And then after that, it will produce our metrics. So let's try to run this and see if it works. Just run the whole thing and see if it works. If it works, if you get something similar to mine on the left, type 1. If you run and get something similar to that, type 1. So on the left, this is now the gene name in yeast. And then these are the samples. Okay. Someone say, why is it indicating characters? Line 12. Let's see here. Let me check line 12 here. Desktop. Yeah. Oh, this is not supposed to be here. Let me get this off here. I think the last one. Thank you for getting that out here. This is not supposed to be here. Thank you. This is not supposed to be here. It's supposed to be 11 here. If that works. We'll try if this works. Okay. Something happens. Just run it. I will fix that later. Something happens here. For some reason, I left something there. Let me don't save it. But run and I'll double check that. Thank you for catching that up. Let's see here. It's annotation file. So the results, let me see here. Let me see. The results should be somewhere. And I think I'm going to stop here. So it's supposed to give us a clean, supposed to give us a matrix. Supposed to give us a matrix. Let me run this again. I'll look at that tonight. Because when I remove that, it didn't run. Something happened, but I don't have time to run it here. It's supposed to give us something like this. Let me see the dimension of this. Okay. The name, the dimension of that. Oops. So we have 6,600 basically these are genes with 12 treatment, basically. I'd like to see if I can find the number of counts somewhere. Because when I did the normalization, then I think it got it better. Basically, what this annotation did was to get us this matrix here. This matrix here. And then from this, from that, we did exploration analysis, like what the other gentleman they talked about. We did PCA, we did I think also did cluster analysis and principal coordinate analysis before we did I did expression analysis. So let me go back to my R Studio here. So to do visualization, there's a script called RNA exploration. So it uses the results of annotation to run that. So I would like you also to run that and see if you can get PCA and cluster analysis for that. So let's run that and we see what we get. Okay, so we have treatment and wild type. So this is a principal coordinate analysis. These are treatment 1, treatment 2, and treatment 3, and wild type 1, wild type 2, and wild type 3. This didn't separate them very well. Let's look at the principal code component analysis. Zoom this. So the earliest principal component analysis separated the two treatment. Treatment 1, 2, and 3 as negative principal component 2, and principal treatment wild type are positive. So it is the principal component to separating the two. Remember when we watched the other movie? It was the first principal component. But for us here is the second principal component. So it's just exploratory analysis. So you can see that at least our experiment work before we do the real gene expression analysis. So the treatment is separated from what? From the wild type by the principal component 2. The treatment 1, 2, and 3 are negative principal component 2. And the wild type 1, 2, and 3 are positive principal component 2. Okay? So try to run and see if you can get the principal components, principal coordinates, and then also I think we have a cluster analysis somewhere. Yeah, so this is a cluster analysis. Let's see here. It's not very good though, but still You can see the treatments as treatment and white type. I don't think cluster will be a good one to use. I would use principal component really, not the cluster analysis. Even the principal coordinate is not very clear. So I would just stick with principal component analysis. For my exploratory data analysis, I would stick with my principal component analysis here. Okay, so for those who were waiting for us in alignment, can you run up to here? After alignment, do annotation after annotation, and then do visualization and see. Meanwhile, I can go and answer some questions here. So first, why is the character empty? Because you don't have the data. If you don't have the data, then it is going to be empty. That means you did not download your data in RNA-seq. So when it goes to read there, when it reads there, it doesn't get any data. So the person here, the number of input file name is different from the number of output file name. This alignment. Did you run the cleaning one first? Because if you clean the data, then it's going to pick up from there. It looks like you are not, the person here looks like you are not getting what? You're not getting data from the clean one. Let me see another one here. Length dimension to not equal. The errors that most of you are getting is because the data, you're not getting data into your environment. So when you don't have data into the environment, then the error will go and read and it will not find anything. So you have to make sure that you have data in your environment. And to do that, one, you have to make sure you have installed your package, because reading the data, you need a package to do that. Two, make sure that your data is in the right directory. Okay? Let me see another question here. I can find. Go here. Q&A. Invalid parameter. Invalid parameter? No counts were generated. Okay, so let me see why here. There should be two reasons why you're not getting this. One is you're not getting the annotation file in DGF format. Two, you not, you don't have the annotation file in the same directory where you can pick it up. So those are the reasons. So it's either not getting the BAM file or it's not getting the GFF file. So somebody asked me, why is ARA so powerful? That's anonymous here. One, because ARA has very many people working. They are volunteers. And two, it's an open source. So people who have projects always come together and do that. Unfortunately, it has a very steep learning curve. And another thing with it is when you're using SAS, you have a problem, somebody can help you. You call them. With ARA, it's your friends to help you, but it's a very, very powerful tool. Let's see, anything else I can. So some will say my BAM file is empty. Your BAM file is empty because one, when you're doing the alignment, you either did not use the reference genome, or two, you did not use the clean data sets. So you need those two. You need the clean data sets and you need the genome, because the R script will always create the folder, because that's the first thing it's going to do. It has to create the folder. Anything else that's come will be after, because the folder has to be created, then the analysis has to be done, and then the results have to be put in the folder. That's why your file is empty, because either the reference genome is not in your RNA-seq folder, or you did not get the clean data in the clean data folder. So all this error that we are getting at, the two reasons. One is the package was not installed. Two is either the reference genome or the data sets or the notation file is not in the right place. That's why we're getting all those really. That's why we're getting all this this problem. Let me see here. Bioinformatics. You see this? You have all this because you are not reading the files. You're not reading the files. That's why you're getting this problem. That's the problem with ARA, because it's a powerful tool, but also, as I said, it has a very steep learning curve, but we should not be discouraged. With practice, we can really do better. So as somebody asked, BAM file. So what's a BAM file? So when you do alignment, you get a format called SAM, which is a kind of text file, but it's very, very big. So what's always done, that text file is converted into binary format. That binary format is easier and it's smaller. That's why it's called a BAM file. It's just a binary format for SAM. So I could have used SAM as an output, but since most of us are using our laptop, I decided to write a script so that you save that file in a BAM format. It saved a lot of space. So I think I think that should answer your question. Okay, let me see if I have another question somewhere. Let's see. So somebody asked me the reference genome should be which file? The reference genome is a file with .fa. So reference genomes are always in FASTA format. So .fa, so FASTA formats are always, it can either be .fa or .fasta. So that's your reference genome. And then the notation file is always either GTF or GFF. Okay? So that's somebody asked that. So your reference genome is the one with a .fa. Okay, so let me see if I can answer somebody else again here. Finish. I think that was Q&A. Let me go back to. So okay, so let's move on. So after doing the exploratory data analysis, now we need to normalize. And the other gentleman explained very well why we do normalization. So right now we are up to here, up to number 3. So we need to do normalization. So what does it mean when you talk about normalization? You talked briefly about it in the video, but I'm going to talk more about it here. Okay, so we really discovered this. We discovered this. We did this. We did PCA. Did this. Did this. Filter and normalization. So there's no need to analyze your genes that are not expressed in either experiments, because when we saw some of them were 0, 0, 0s, that's one reason. So sometimes you have to remove those ones. So because yeast has around 6,000 genes, but some of them are not expressed at all. So sometimes we need to remove those. That's one reason why we do filtering. Okay. Another thing that we need to do is sometimes when you're doing filtering, you have to normalize so that you have your file in a certain format. So we use when you're using HR, but now I'm going to introduce HR here. HR is very, very commonly used in RNA-seq, but sometimes it's hard to use because of the type of the file that you're using. So it's always advisable when you're using HR to have file read in what? Demulated format. And then also HR stores its objects in what is called DG list. So if you get your file, you have to convert that into DG list. So sometimes when you're working with HR, you get in such problems, okay? So when you're using HR, it helps you to remove those counts that maybe are missing or that are very low. And the end product is what? Is the count matrix, okay? So like for our case, after filtering everything, you'll find that the last file that we have, it will not have zeros and one, okay? So normalization. So the first one was filtering. You filter those that are zero, you don't need them. But now you have to normalize. So this one was explained in the video very, very well. What I'm going to do, I'm going to go further and explain to you there's a certain type of file that sometimes when you send your sequence, your sample to be sequence, and most of the time they send you back in a matrix in either Excel or comma separated file, and they have like different values. So we're going to talk about that. So this depends on the library size and also on the gene length. Okay. So I'm sure people who have been working with RNA-seq data always, when they receive their data, the data is one of these formats here: FPKM, RPKM, and sometimes they don't even explain to what they are. But those are always normal data that has been normalized. So what's FPKM and RPKM? So this you divide by both the gene length, you compare across the genes in the library, okay? And then you have the fragment per kilobase of transcripts per million mark. So these are mainly used when you have the sequencing in pairs, okay? But RPKM always is used when you have single read, okay? So these are the two that are most commonly used, especially when they're working with the people who don't have bioinformatics skills. They always send them this type of file format. And the last one is called transcript per million. So this is done by adjusting the length so that the sum of all normalized genes are equal to 1 million. Okay. So when you have this, after normalization, then be ready for your what? For your doing your gene expression analysis. So after alignment, you do annotation, you do normalization, and then the last part is you do what? You do your the gene expression. So what I'm going to do, since we're running out of time, I'm going to answer some few questions so that tomorrow we start with gene expression and we finish, and then we move to genomics, okay? So I would like to stop here, and then the next 10 minutes, answer questions. Because if I go to gene expression, I think will be better. I start gene expression tomorrow. Okay. So for the R script, let me show you here. The R script that we managed to use today, we installed the packages. We got data from our computer, we loaded into R Studio. We did the checking. We discovered that the first 15 nucleotides were not good, but the rest were good. And then we also checked for Fred score. We found out that the Fred score were good. All the sequences were above 30. So we didn't do anything at all. So that was after checking. And then the third script, we did the cleaning. We removed the first 15 nucleotides from all our reads, millions of them. And then after that, we save those in a new folder, okay? And then we use this script here to check the clean sequence. And indeed, we found out that the first 15 nucleotides were removed. They were removed. So we're very, very sure and confident that, okay, now the clean sequence, now we're going to do what? We're going to use those for the next step. And the next step was to align those reads to the genome. And our genome is the yeast genome, and the file format is FASTA. So we use this for alignment. We align our reads to the genome. And then after that, we got our alignment in a BAM format. A BAM is basically a binary format of SAM, but SAM the file is too big. So if you can make it in a BAM format, it's easier to store, especially when we're using our laptop. Then after that, we did the annotation of all those reads into genes. So how many reads per gene were expressed? So we use this script here, and the file that we use for annotation is GFF3 file. And after annotation, we got a matrix. In the matrix had had rows of genes, and then the columns were the samples. And our samples, we had 3 treatment and 3 wild type. Wild type means check or control. And then in each column, you have the number of reads for that particular gene for that sample. But as we watched in the movie, in the video, we had to normalize, because it doesn't mean that if you have 300 reads for that sample for that particular gene because of the expression level, because sometimes of the what is called the calling or the machine. So what we do, we normalize. But before we did that, we had to do the visualization. So we did principal component analysis, we did cluster analysis, and we did we did principal coordinate analysis, sometimes called MDS. But we've realized that the best was principal component analysis. That's why you have to do more than 2 or 3 methods. So we realized that the experiment was good because when we did principal component analysis, the 3 treatments that were treatment and the check, they were separated. They were separated by principal component 2. The treatment were represented by negative principal component 2, and the wild type were represented by positive principal component 2. And then the last thing that we did was normalization and filtering. So the filtering we did was remove all those sequences that were not, genes that were not expressed, because they were all zeros in either treatment or control. We removed them. And then another thing that we did was normalization. So with normalization, we did by the gene length and also part per million. So that's where we are stopping today. And tomorrow, we are going to do gene expression analysis statistically, because all from number one, from number 1 to number 8, basically, you're preparing for the last 2. Because this gene expression analysis, you're now going to use statistics to determine the difference between wild type and the treatment. And then that's analysis, and that's why you now do the enrichment, like gene ontology, the KEGG. So we're going to finish this tomorrow, and then we'll go to our genomics, okay? So at least this way we are stopping for today. We started from here up to here. So the next 5 minutes, I'll answer questions and we'll call it a day, okay? So the problem here is directed to the FASTQ files, okay? So the FASTQ files are supposed to be under RNA-seq. So that's where they're supposed to be stored, okay? Why are we setting the working directory? We're setting the working directory because when you're running your script, it's going to look within that directory. Okay, thank you. Thank you, I appreciate that. Let's see here. Okay. So we're gonna get this again. See here. Any alternative way to import the FASTQ files? Okay, I will show an alternative way tomorrow. Maybe you can use another package. Okay, so I'll bring that tomorrow. What should I do from receiving error frequently? Although I've been using the steps you have. Yeah, this one just have to practice. The biggest problem is the directory, really. We'll repeat that tomorrow. But before you close, is there any other way to run it fast? I mean, we could run this fast, but some of this takes time. It's a workflow. Remember when you were installing the package, and some of you just when we were working with peace and installing the packages, and some of you ran straight away. So some of them were skipped because you put A for all and then take some time, sometimes 2 or 3 minutes. So that's why you have to go step by step. But sometimes when you don't have, especially when you're installing something from the web, it takes longer. So sometimes you have to wait for this to run faster. Another thing, how it can run fast is when you're using servers, because what we've been doing here in servers take minutes. But here we're working with that. I think most of us are working mainly with our laptops, really. Okay. So I have 2 minutes left to answer some of these questions. Let's see. Go back to Q&A here. Okay. Let's see. What should I do? Assuming the error frequency, although I've been using these steps earlier. Yeah, we need to, I think this again it has to do with the package or where the files are. So it's coming to 5. If we have anybody from the forum can close.
Yes, so, this wonderful session. We really appreciate colleagues. If you have any questions, we told you platform where you can inquiry. And if you want to replay the training of today, you can visit our YouTube channel, Forum Network. For today, let's stop here and we meet again tomorrow at the same time. Bye.
Bye. Thank you.